All right, so welcome everyone to Shared's COVID Q&A. We are obviously so grateful for the 4 panelists that are here to give their time. We have Dr. Angela Haczku in the top right there. We have Miss Renate Hallock on the second picture right there. Dr. Walter Leal, third picture, and Dr. Jacob Alexander. So before we get started, I'm just gonna drop a few links in the chat. So the few links, the first one is if you want to ask any of the panelists a question. So we already have 6 questions prepared for them to answer, and we're gonna be going through them. But if you have a question of your own, please go ahead and fill out the top link, because we do want you all to be able to ask a question. And also if you feel like you want to unmute yourself and ask a question, we also obviously want to have that happen as well. But the form is there if you don't want to unmute yourself. And then the winter application for Shared Club is also there, and then our Discord as well. So let me go ahead and go through this. And then Dr. Angela Haczku, if you would be able to introduce yourself, please.
Yes, I am delighted to participate in this panel. And thank you for the invitation, both Ishita and Alexander. It's an honor and very exciting as well. So I am a professor of medicine at the UC Davis School of Medicine. I'm an MD PhD, and by training, I am an immunologist and work in the field of pulmonary immunology. So COVID-19 is right up in this area. I also am the Associate Dean for Research for the School of Medicine, overseeing day-to-day operations of the research enterprise. That was before COVID. And after March last year, I was in charge of overseeing the medical school research endeavors targeted at COVID-19. And it's a highly translational area, meaning that basic research has to be connected with clinical approaches and clinical sciences. And also it has been a really exciting whirlwind of collaborations, because literally research results drive clinical operations and clinical practice as well. So it is medical discovery in the making, and seeing it live. It's a very exciting time, as scary as this disease is. So I'm going to stop here again, and thank you.
Thank you so much. Dr. Alexander, would you like to introduce yourself as well, please?
Sure. So my name's Jacob. I'm a physician, an internal medicine trained hospitalist. So what a hospitalist is someone who only sees patients in the hospital, adult patients. I work for Sutter Medical Group, which covers Sutter Davis, downtown Sacramento, Roseville, and Auburn. Although I've worked at all 4 sites, I mainly work at the Davis site now. I've been there for over 2 years now. I did my medical school at George Washington University in Washington, D.C., my residency at LA County, USC. And now I'm here. I've been seeing COVID patients pretty much since COVID came to our shores. As a hospitalist, we see, I'll get into this a little bit more, but we're typically the providers, the practitioners who see COVID patients when they come in. But anyway, I'm looking forward to this discussion and answering all your questions.
Thank you very much, Dr. Alexander. Dr. Leal.
Thank you. Thank you very much. My name is Walter Leal. I'm what we call the distinguished professor in the Department of Molecular and Cellular Biology. As you get older, the title gets increasing, and they don't have another word, so they put a distinguished professorship there. One of the reasons that I might have been invited here is because when the pandemic started, I started to organize a series of COVID symposia, and it was very well attended from people from overseas and things like that. So I'm not a specialist. I'm not a virologist. I'm not a vaccinologist. The only thing I know about vaccines is that they don't work very well if they stay in the freezer. They only work after they've been injected on people. So let's discuss and also talk a little bit today about some other aspects of our research and things like that, and how COVID has been affected us here at the research at the UC Davis. Thank you. Thank you for the invitation.
Thank you so much. Thank you. And then finally, Ms. Hallock.
Hi, my name is Renate, and I thank my daughter Carolyn very much for inviting me here. I'm a radiologic technologist, and I've been working for Kaiser Permanente for the past 15 years. The first 5 years I worked in general radiology, just taking x-rays of broken bones. And then I became really interested in the more technical aspect of what else x-rays have to offer. And I found out about the subspecialty interventional radiology. And I don't know if you know what that all means, but it's really, we do minimally invasive procedures under live x-rays. So not just static x-rays, but running x-rays. And we use contrast dye in order to make the body parts really visible so that we know what we're doing. Our bread and butter really is, I would say, for example, putting chest ports in so patients can get chemotherapy. We do work a lot with patients who have end-stage renal disease and the kidneys don't function anymore, who have now grafts and fistulas. And these vessels can get blocked. And what we do is we take little tiny needles, go into the blood vessel, inject even tinier wires and catheters, and then open up those blockages with balloons. And what we do in Kaiser Richmond, and we're the main hub for Kaiser Permanente in Northern California, we have liver cancer patients and we deliver radionuclides. It's called yttrium-90, specifically to these liver tumors, and are very successful then with that and make them shrink, or we had some patients who were completely healed. We see, since COVID started, we see, of course, COVID patients, and things have changed for us a bit when it came to how do we protect ourselves and how do we screen patients before they come in. On some of the patients, for example, gastric tubes, we have to make sure they really have, they don't have COVID, or if they do, we need even better things to protect ourselves.
Thank you very much. So as you can all see, we have a whole variety of healthcare professionals, researchers, professors. So somebody wants to come in. Perfect. So we're really fortunate to have a whole variety of backgrounds, and let us go ahead and get started with the first question. So the first question says, introduce yourself and describe your position and daily work before COVID. So obviously most of you introduced yourselves, but can you give a little bit more of a description about how your work was before COVID and what sort of work do you focus on so that people can better tailor their questions towards you? Let's start with Dr. Haczku.
Yes. Before COVID, 50% of my effort is devoted to my laboratory research. As I mentioned, I'm a respiratory immunologist, and I run a research laboratory with students, postdoctoral fellows, and senior specialists. Our area of interest is pulmonary disease, and we model these diseases, especially inflammation and how the immune system directs inflammatory changes in the lung. We model these diseases in animals as well as in vitro, and we also conduct human studies. In vitro, we focus on the airway epithelium, also the target cell of entry for the COVID-19 virus, SARS-CoV-2, just mentioning that. And the disease modeling in animals is specific in non-human primates. We have one of the world's best non-human primate research center here at UC Davis, and it's very suitable to study diseases that are shared between these animals as well as humans, because we also share about 85% of our genetic information, the genetic code. And so symptoms and disease mechanisms are very similar between humans and these animals, so these are perfect to model diseases in. The other 50% of my time, as I mentioned, I direct and oversee research operations of the School of Medicine. In this, we look into how we can help researchers in their daily endeavors. And maybe later I can tell you a little bit what work we did after COVID-19 broke out, because it completely turned things upside down. And that was a very interesting process.
Definitely. Yeah, we definitely want to know about what happened after COVID. That's actually the next question. It's like a foreshadowing of it. But Ms. Hallock, what about you and your position? What have you been doing before COVID?
So you have to imagine our procedure room really like an operating room. So we work under with sterile procedures. We are scrubbed up just like an operating room. And of course, we have to work under rules and regulations to make sure everything is safe. And we usually see about 5 to 6 patients a day. We are open from 8 in the morning, and the last patient we usually see at around 3. Sometimes we might get an emergency case, somebody who has some GI bleeds, and that is just an emergency, and we just can't go home. We have to do it. But around, I would say, 6 patients. And it's really hard to say before how it was before COVID, because in a way, not so much has changed. It was just a little bit, we had to find ways to better protect us. So what I do in the morning, I get ready for the first case, set up my table, patient comes in, and I stand right next to our interventional radiologist, and we put the patient on the table, and depending what kind of procedure we do, we scrub the patient, make him all sterile, put on a drape, and then we work on the procedure using either wires, balloons, catheters, depending. We might put an abscess in. So let's say there's a patient who has, for whatever reason, maybe because of cancer, just because the body does weird things, has an abscess, we put little drains in, use X-ray to find the abscess, find out how big it is, put in a drain, and send the patient on their way. And then we get ready for the next patient, send those images across we have taken, and get ready for the next patient.
Great, thank you so much. Dr. Leal, what about you? How has your research been focused on prior to COVID?
All right, so prior to COVID, I used to work every day. Now after COVID, I'm working only 7 days a week. And the focus of our research is on insect olfaction, how insects perceive the world in terms of smell. And specifically, we work with mosquitoes. So there was a period of time when they stay at home, order came, the university shut down, basically. That was the most difficult time because we have here a colony of mosquitoes that started in 1940s, way before I was born. And I keep this colony as a property of the University of California. It's a very important resource that is, we can do lots of research on that. And that was my major concern during this time, how we can going to keep this colony alive, because these mosquitoes need blood every day. And it was a little major problem there. The other part of the research that we do, we were able to slow down, stop at some time, and now after the university came back to the phase 1 research, we were able to resume. But the major effect that had on us is that we deal with lots of overseas students or visitors or faculty that are on sabbatical and things like that. And these people are not going to be able to move to the country and outside of the country. So we had students here that had to stay longer because they could not go back home and things like that. And the others that were coming and no longer came. And so the research has suffering a lot, but we are being able to keep the colony alive. We could be able to keep the research going. And again, we are interested in how mosquitoes find people and how we can repel these mosquitoes, and also how can we find place and smell that attract mosquitoes to a certain place for lay the eggs for oviposition. And with that, we trap mosquitoes that are infected. Because right now, COVID is the major problem, but the other disease did not stop. Zika transmission by mosquitoes, dengue, and so on, and the West Nile virus here and the North California, everything is going on as COVID is the major problem, understandably. So in a big picture, affect our research, but we are surviving.
Good to know. Yeah, I'm can't imagine how much it's actually affected research going for that, because it seems like most of the research now is based on COVID. So, I mean, at least you're still able to continue on with some of it. Dr. Alexander, what about you? How is your work prior to COVID?
Sure. So the day-to-day functioning of a hospice, essentially, you have a panel of patients that ranges anywhere from 10 to 15. And you what's called round on those patients. You check their vitals, their labs, overnight events, how they're doing, whether they need additional tests ordered, whether they need to go to interventional radiology to have a drain placed or a special procedure done, and then ultimately discharging them home. So before COVID, it was day to day, it was not too stressful. You build great relationships with the patients. You try to sit at their bedside, really talk to them, get to know what's going on, spend a lot of time collaborating with other specialists in the hospital, people like case managers who work on discharge planning, social workers, respiratory therapists. So it's a lot of interaction with others, which is a really fun part of the job. And then on top of that, you would do admissions from the ER. So people who needed to come up to the hospital, you would go down to the ER and admit them. And I can in the next segment elaborate on how that has changed since around this time last year.
Perfect. And then, yeah, so that was leading sort of directly into this question, which is, how has COVID-19 impacted your work and or workspace? Do you work directly with patients that have COVID? So I think Dr. Alexander, since you were mentioning it, do you want to go ahead and take this one first?
Yeah, sure. So I was, saying how, a lot of what a hospitalist does is, face-to-face time interacting with the patient, specialists, discharge planners. I'd say that was the most profound impact. Initially, there was a lot of people started working from home, or case managers were just working in their offices. So we didn't have that face-to-face time. There was certainly concern about getting close to a patient. So, having to wear a mask, having to wear a face shield certainly puts a physical barrier between you and the patient, which I think makes a big difference in terms of the patient's overall perception of their hospital stay and the interactions they have with the providers in the hospital. Certainly fear, concern, I'm sure a lot of other nurses and respiratory therapists had about contracting the disease from a patient. If you remember in the beginning, tests would sometimes take 3 or 4 days. There wasn't good guidance in terms of wearing an N95 mask versus surgical mask. So it was just a lot of anxiety, a lot of concern, especially in that beginning period of contracting the disease, bringing it home to the families, and and then of course seeing a whole new disease process, which we were really unfamiliar with. There have been tons of studies about heart failure and diabetes and how to best manage certain skin infections and how long you're supposed to give a patient antibiotics, but we didn't really know much about COVID. And so we were, and this was going on obviously all around the world. Practitioners are having a really hard time finding out how to get these patients better, how to keep them alive. And certainly we've made progress, yet I would say even till this day, there's some times when we feel really helpless when these patients get so sick, and we have such few resources at our disposal, such limited medications that we're able to use and that have been shown to be beneficial, that even a year after, as much as we learned about this disease, how it's transmitted, how it works in the body, how it attacks the, the lung cells, we still, I feel like, have a pretty long way to go in terms of really being able to effectively treat and manage these patients.
Thank you, Dr. Alexander. And sort of on the same lines, Ms. Hallock, so based obviously on this question about how it's impacted you, but there's also a question for the audience that says, this virus has been horrible for so many people. Have you been able to find any silver linings personally? So obviously, how it has affected you, how COVID-19 has affected you, but also have you been able to find any silver linings?
I definitely feel what Dr. Alexander felt, that in the beginning we were totally scrambling. I mean, you would think you work with in a hospital field, you're so kind of guided by rules and regulations, you know how often to change your mask, and out of a sudden we didn't even have N95s available. So out of a sudden we were told you have to wear that N95 for a month or even longer. And you knew this cannot be safe. You looked at it physically and it was just a dirty mask, but there was just nothing available. We were scrambling on all ends and tried to make it work. We were then of course coming home and yeah, taking a shower, trying to keep our family safe. So I think we were just, I felt like this was my first day on the job, really. But then we were scrambling a bit, trying to find more rooms in case we had to expand our ICUs. So what we had to do is, which was a little bit of a tough decision, we had to really cancel patients for elective procedures. For some of the procedures, that is in a way easy to do because it might just be a little bit of like a cosmetic procedure, let's say. But we have a lot of patients who need dialysis and they need their vessels checked. But we knew we can't do that on a regular basis anymore. We can just see is the vessel still open. So we had to postpone these procedures and then realize these patients would get very sick after a while, and out of a sudden their vessels were not just tight, they were blocked. So we ended up having that problem. So then we realized, okay, we have to open up back business again. But what do we do if out of a sudden the ICUs fill up? So that was, I want to say, a couple of months, 3-4 months, where we really were in the spot where we weren't sure. But when you ask about the silver lining, what I found was really interesting. And I came to interventional radiology because I knew this would be such a great tight team with doctors, nurses, and technologists. That's what I liked about it. But we became even closer because we felt we were all in the same boat. We found out things about our personal lives you would have never admitted before. But we were so, it's almost like there was a different layer of our personality. And I have to say it, and it sounds really maybe strange, but I think we all enjoyed that in a way. And I don't know how you feel about it, I mean, the rest of you, but I'm sure you can somehow share that experience.
Well, I'm sure. And there was also a follow-up question for you that someone asked. They said, this is sort of a long question with multiple parts, so whatever, stands out more to you. It says, have the pre-procedures changed because of COVID? Do patients need to get a COVID test before the procedure? How do you keep yourself and your team safe during the pandemic? And do you use PPE? Do you use more PPE? Is the environment of the room different? So sort of a lot of questions for you in that one, whatever, stood out more in that, if you'd be able to answer that one. So
fortunately, getting enough PPE is not an issue anymore. So we can change our N95 after each patient if we have to. Pre-procedure wise, we have certain procedures which stand out. So some procedures, they can just cause aerosols. For example, if you have a patient where we put gastric tubes in, that just putting a gastric tube in or putting a nasal tube in can cause just patient to cough. That, of course, if that patient now would have to happen COVID, that would be a big deal. So what we decided on those cases, that they would all get a COVID test before. So before this patient even comes in the room, if they don't have a COVID test, we would not be able to do the procedure. But having said that, there are emergencies where we don't have time. So we just have to then, double up. So we put the N95 on, we put the face shield on. I mean, that's something we have done anyway, but it feels like it's a little bit more. And if we're just not sure, does this patient have it or doesn't have it, we just assume the patient has COVID. So that has changed. And of course, and that's probably also when we wash our hands, even though you're supposed to wash your hands all the time, I think we wash it even more. But that, I think that became sort of a habit. But I would say another problem, what I see in interventional radiology because of the COVID cases, that room really needs to be turned around quickly, but at the same time, we have to make sure we clean out that room. We put a light in there to clean out, and that just costs, it's just extra time. So we have to rush even more inside the room. At the same time, trying to make it safe and make it a safe environment for us and of course the patient.
Thank you for answering all those myriad of questions. The next question actually focuses on research. So I wanted to bring in Dr. Haczku and Dr. Leal. So let's start with you, Dr. Leal. How has COVID-19 research developed since its outbreak, and what is the current research focus?
So again, my research is not on COVID-19, but it's on mosquitoes, and it changes dramatically, like I explained before. But what I would like you to share as an opinion, and is the importance of science, because the way that we developed this vaccine, for example, so rapidly in a short period of time, less than 1 year, the vaccine from the research bench to be available. Unfortunately, the next step has been a failure, but the research part was a tremendous success. And the reason for that is because people have been doing basic research before. For example, they have been studying SARS-CoV, the coronavirus before that one. Let's say that the people in Congress or someone normally criticize people doing science, and they would look back at that, they would say, oh, this is a waste of money. This SARS is not important at all. Now it became so important because that foundation, that's what helped us, or helped the scientists in general, to discover what are the receptors and what kind of target can be made for vaccine and everything. Because if you had to start from the zero at that point, we will still be working on these details 1 year later. Today, where we have a first year anniversary of the COVID starting in the United Kingdom, and that's it's amazing that 1 year is there, but the research was able to put this vaccine together because we have been doing fundamental research. And the people doing fundamental research, basic research, sometimes get attacked by people in Congress. They don't understand anything, and they just say that, oh, this is a waste of money, and sometimes they are even targeted for that. So I want to emphasize the importance of research, fundamental research, that brought us so rapidly to a vaccine.
I know that it's not your specialty per se, but would you be able to speak more about the research of mRNA vaccines, since it's sort of a new type of vaccine? Or Dr. Haczku, I see either one of you.
If you
I
don't,
think you're muted, Dr. Haczku.
Sorry, I apologize.
There we go.
My friend, a Hungarian scientist, also Dr. Katie Kariko, we worked together at the University of Pennsylvania, and she discovered the mRNA vaccines, both that are used in the Moderna and the Pfizer vaccines. And just like Dr. Leal explained, her mRNA research has been sidelined for decades. Basically, she devoted her entire career to this, and she got very dispirited because nobody believed. They said, you cannot do anything with mRNA. It's a very inflammatory molecule. It gets degraded right away as it gets into the body, and you can't use it for anything. It's useless. Your research is useless. But she remained adamant, and she worked out a breakthrough that made the mRNA useful and not degradable when it's injected in the body. When she teamed up with an immunologist also at the University of Pennsylvania, I worked there before UC Davis as well, and they patented this, and so this patent got then bought by Moderna as well as BioNTech, where Dr. Carico is working now. And because of decades of prior fundamental research, in fact, indeed sidelined fundamental research, the vaccine industry was able to jump onto this technology, and that is the sole reason that millions of vaccines were able to be produced, and injection could start in December last year. Otherwise, it would have never happened. So thank you, Dr. Leal, to bring this up.
Another important element is that the people studied these proteins before, the spike protein before, and they knew that you can generate only the antibody. I'm going to be discussing this in my class, in my biochemistry class, actually. So the protein has the binding part of the protein, right? The receptor binding domain, we call it. And this protein comes is a three units. And one unit is of that bind sticks out. We call it the up and the or open state. And the other two stay in the closed state. And they discovered this thing way before. And now the researchers were able to design this spike protein in a way that this bind is always in the, one of them is always in the up position. So therefore they could design the antibody that match. Because if they didn't know that, they didn't know this fundamental information, they would not be able to design a vaccine that would be useful, because the virus would hide the receptor all the time. And now they were able to make the receptor. So this spike protein that is in the vaccine is somewhat identical to the spike protein in the virus, except that there's two amino acids that are different, two proline amino acids they put there, so that when the RNA, like it was explained now, when the RNA is made, survive and transcribes into protein, the protein has this site sticking out, so the vaccine is going to be effective. These are important information that the public does not appreciate if you don't tell them. We have a very hard problem with science because we don't communicate well with the public. We don't tell what's going on, what's important. And like was just explaining, this research that was surviving and they being alienated in a department because no one believed this RNA vaccine was going to work. And they modified the nucleotides. So therefore, it's a stable and it allows the expression of the target protein. So research is fantastic. We waste a lot of money. Yes, we waste a lot of money because none of things that we believe is going to work doesn't work. But the one that work pays off for everything else that was lost and more.
And if I may add just another very tangible way, as I mentioned, that research in COVID-19's case directly drove clinical improvements. For example, testing. So you may all aware that the first community-acquired COVID-19 case was discovered here at UC Davis, and that patient was in a very dire situation when got into the ICU, and the doctors, Dr. Cohen and his team suspected that it could be COVID-19, but they couldn't come up with the diagnosis because at that time, entire Sacramento County was allocated 24 CDC kits that were approved by FDA to make the diagnosis of COVID-19. And because this patient did not travel to China or was exposed to non-COVID-19 persons, was not deemed eligible for testing. So it took perseverance and many, many phone calls by our ICU doctors for the authorities until they approved it. And it had to be done in a very short period of time. And as soon as the patient was identified as indeed being positive for COVID-19, she received remdesivir, which is still a controversial treatment, but if it's applied at a right period of time in the ICU, it can actually significantly improve the patient's state. And we believe this what happened to this patient as well. And remdesivir was also discovered here at UC Davis by Niels Pedersen, a veterinarian researcher who researched coronaviruses in cats and found out several years back that remdesivir cured some of the cats of the coronavirus disease. So that was one thing. The other thing was that after the patient was identified, the virus was isolated from this person and propagated in the virology labs in the primate center on the Davis campus. And our laboratory clinician, Nam Tran, was able to develop an in-house assay that was within the period of 2 weeks. And by that time, FDA approved in-house assays to test COVID-19 in patients, and also the CDC laxed the criteria of testing, and they said anybody who is reasonably suspected could be tested, and in-house tests for diagnostic tests could be used because there weren't enough tests. So as the researchers were identifying the virus, those data were immediately applied to develop clinical testing, and as well as different treatment approaches for COVID-19. So in the position, you also asked about the silver lining. So the silver lining for me was the eye-opening situation when researchers and clinicians were enthusiastic and able to work hand in hand and apply the discoveries immediately to the actual problem. And I think this attitude should be reinforced and fostered and supported in any ways, because that can move medicine forward much faster than it has been moved so far. So that was my silver lining. Thank you for that question, Alexander.
Yeah, thank you both.
If I could add, Alexander.
Yeah, of course.
Angela mentioned about the testing situation 1 year ago, how ashamed we were at that time that we could not do a qPCR reaction with something simple that everyone does in the lab. Anyhow, so now 1 year later, yesterday, the New York Times had 1-page article about the Davis and the city of Davis that are doing this health Davis together, that anyone in this city, anyone in this school can be tested at least once a week with a rapid test developed here at UC Davis. So this is marvelous. 1 year, and we are here now in the situation that test is not an issue. So I hope that pretty soon we are going to be saying the same about vaccines.
Yeah, of course. So based off of what you both said, I think 2 questions from the audience would be a good thing to ask right now. So the first one could be for anybody. It's good. So they're asking, what's a good approach towards people who are skeptical of COVID vaccines? So if any of you would like to take that question.
So I just very quickly, I am actually horrified by the amount of false information and misinformation circulating in social media as well as on the internet, and even by newspapers, and that's really scary. In regards to vaccine hesitancy or anti-vaxxer movements. Vaccines are considered to be the greatest breakthrough in medical treatment in the history of medicine. They actually improved life expectancy of humans by decades, and quality of life of humankind exponentially. Also, vaccines are the safest kind of medication. Why? Because in a very tiny amount of material that is being inoculated into the person, it starts basically just harnessing our natural immune system to boost the immune system and focus the immune system against the invader microbe, let it be a bacterium or a virus or any other agents. And so these are very simple facts that any vaccine hesitant or anti-vaxxer person need to consider. Also, once vaccinated, you not only protect yourself, but you protect the surrounding of yours. You protect your family. So think about that when you don't want to get your children vaccinated. You know, what kind of suffering you are exposing them to, or if you don't want to get yourself vaccinated with COVID-19. And thirdly, herd immunity is a status of immunity that would protect humankind against nasty, nasty viruses like SARS-CoV-2. So I think these are three very simple messages that should be propagated by everyone who is at present here, and try to make sure that that everyone understands it, and there is no emotion, no politics, nothing else involved. These are pure, true, objective medical facts. So that is all I wanted to say.
Was there anyone else on the panel that wanted to answer that one as well? Dr. Alexander?
Yeah, I would just say, try and focus your time on the people who are skeptical rather than the people who are firmly anti-vaccination. If they hold, harbor feelings about all vaccines, you're probably not going to change their mind. Should really, try talking to the people who are maybe skeptical of the quick turnaround of the vaccine, side effects. There are a lot of headlines, unfortunately, you hear about people who died hours after getting the vaccine. That's going to scare people. And I think what you just have to show them is, the article that was published in the New England Journal of Medicine showing the incredible effectiveness of the virus, it was 1 or 2 weeks after the second dose. You talk about how diseases have, been almost completely eradicated. Polio is a good example through vaccination. And, rather than stigmatizing them or try to challenge their beliefs with positive, reinforcement. Show them that you're concerned about their well-being, that they're not only like Dr. Haczku said, protecting themselves, but protecting the people around them. And I think at least I've had success that way in maybe reassuring people, calming them down about their fears about this vaccine.
And I believe I found the New England Journal of Medicine that you were referring to. So if people want to check out that link. There was another question. I think Dr. Leal, you might be the best equipped to answer this one. Let me find it again. It was, how effective are the saliva tests and what is the most effective COVID test?
So about COVID test, we have to repeat this thing every time, right, Angela? There are two different types and they look at the two different things. One is the PCR-based test that are looking at the RNA itself. And there's a curve of a type of the curve of the RNA that it happens, let's say, during 10 days. There are some exceptions, but during 10 days of the disease, that they are going to be able to find this RNA, and therefore for that a PCR-based test, like the saliva test is one of them. After the disease kind of disappear, then the antibodies appear in there. So if you are going to look at serological tests, then we are going to find it then after sometimes the manifestation of the disease. So if you are going to do the serological test in the beginning when the person is sick, it's more difficult to find the antibody because they are still at a lower level. Or the reverse of it, if you are late after the disease, you are going to try to find it by qPCR, we are not going to find any trace of RNA, except in very special cases. For example, one of the first patients here that was treated at UC Davis, she keeps shedding the virus for months after she was sick, and it was still negative, but was shedding the virus. Anyhow, so my point is, what's the best test? Depend of the time. Right now, everyone is looking for asymptomatic people because they spread the disease and they don't even know. So that for that kind of people, qPCR-based tests. And for something later, if the vaccines work or if you have immune or other things, then we could look at the serological test. Right now, qPCR-based tests, and the good thing about the saliva test, because people who took the other test before the swab probably remember how hard it was. I mean, to put that swab inside, there's some very uncomfortable. And now with the saliva test, anyone can spit, and it's so easy, so accurate. Of course, it's not as accurate as a regular test that takes a long time. But one issue is time, another issue is accuracy, and we can pay off a little bit by having more results and more rapidly. Back to the issue of vaccine, I don't want to go too much on this. Dr. Alexander put very nicely. There's a group out there that we are not going to convince. You can show any science to them. We're not going to change their mind. This was a great point, to talk to people that are in between. They still don't have enough information, but they see both sides. And I can understand that. One thing that I will tell them, if they are in front of me of the line, give me a spot because I will take that vaccine immediately. But a little bit more serious about that is that they should understand that yes, there might be some discomfort, there might be some side effect. I was hospitalized only once in my life when I was in Japan. I was a student in Japan, and I got shingles. Okay, so I had to be hospitalized. And now, this many years later, the doctors tell me that there is a possibility that they're coming back. So take the vaccine. I took Shingrix. To me, it was the worst vaccine that I ever had. And the Shingrix vaccine took me 1 day after the vaccine, and it's 2 doses as well. And 1 day after the vaccine is horrible. You don't feel good, blah, blah, blah. But I will exchange any day like that after the vaccine for 30 seconds that I was in the hospital. Even more so with the case of COVID. One thing is that, oh, you don't feel good 1 day, or the arm is sore or something. But remember, people out there are getting intubated to be able to survive, and many of them do not survive, and a number of them remain sick for a longer period of time. So do not hesitate. This is your opportunity. The science is solid. There is no things like this is going to be incorporated in your DNA. That's impossible. RNA be incorporated on your DNA. This, like Angela mentioned, one major issue was that to make the RNA stable enough for a short duration of time that will be able to transcribe in the protein, because the body tends to get rid of this thing immediately. And for some people out there to speculate that this is going to be incorporated in your DNA, and even this crazy idea that the government is going to put a chip out there. So we can't discuss it in a scientific basis. This completely disinformation, and as a scientist, we feel very disappointed that it is going that way. But unfortunately, that's the world that we live right now.
Dr. Leal, just a quick follow-up on that, since I know that some interesting headlines have been happening with what's going on in China with the rectal probes. Is that something that you could see happening in the US? Is that something that's actually viable?
To be honest, I got a link to that information, and I didn't have time to read, so I'm going to skip that one.
Does Dr. Alexander, anybody? All right,
No,
we'll go on.
We have no plans to do those at Celery, at least as far as I'm aware.
That's what I thought. Let's go on to this one, because there was also a follow-up question that someone asked in the question form. So this one says, what are some effects of COVID-19 that you think will stay with us during the next couple of years and beyond? Ms. Hallock, would you like to start off with this one first?
Yeah, so I mean, I would think for the next couple of years that we, not just in the hospital, just in general, I think masks definitely will be there for a while. Same as shielding. Let's say you go to a grocery store, you see the plexiglass in front of you. I'm pretty sure that will stay. And I mean, hopefully the hand washing will stay for a long time, because I think that should be done anyway. And I think we learned from this. I think just in general, we will be more careful. What I think will be another effect, and I was just thinking about it since, I mean, I have kids, that when I watch little kids, that there's always already that hesitancy to get close to a person, and they put their masks on, and that could be, I think, kind of a negative effect. So I'm not so sure how that will affect their psychology, but I think that will stay with us for the next couple of years.
Anyone else want to take that question as well?
So I just would like to add an interesting historical perspective. Another Hungarian scientist way back in the late 1800s, early 1900s, Semmelweis, he actually invented the hand washing. Believe it or not, up until that point, hand washing wasn't a thing. People didn't wash their hands. They weren't particularly clean. Doctors didn't wear white coats. They wore black coats because blood and other excrement wouldn't show up badly on the black coats. So that was doctors and nurses' uniform and healthcare workers' uniform at that time. And puerperal fever, which is the disease that mothers who are giving birth would get, was rampant in hospitals. So mothers would rather, if they could afford, hire midwives to come to their homes to give birth rather than go to the hospital, because it was almost equated with death. And Semmelweis figured out that this puerperal fever could be transmitted from one person to another by palpating the moms in labor by the doctors, by the healthcare workers themselves. And he requested the hospital, that was the very famous university hospital in Vienna, to wash their hands. And just like researchers these days, as it was mentioned, he got completely sidelined. In fact, they kicked him out of the faculty, saying that he's insane and he talks rubbish. So he implemented his guidelines of hand washing first in Budapest, and puerperal fever prevalence went from 30% down to 1 or 2%, which was an amazing result. And actually, eventually, the hand washing was adopted and accepted, and now bathrooms are put into restaurants, and it's, you see the little labels say that employees must wash their hands before they. So that took about 100 years, and now I think other things will change, and life will never return to the way it was before, after COVID, because we know how the mask can protect aerosolized infections. So I just feel that life as we knew it before last year might not ever return, and then there will be some permanent changes that we are facing now.
Thank you so much, Doctor. Seeing that we only have a few minutes left, I just wanted to go on to the last slide and have you all give any final remarks, any final advice, anything like that. If we could start with you, Dr. Alexander, any final remarks from you?
First of all, I'd like to apologize if you hear my daughter and the dog in the background, but I would just say, you know, all of you as pre-medical students or whatever, you know, pre-health field you're in, you know, you can work really on being an advocate. I think the biggest fight we have now, like we talked about, the fight against misinformation, getting people vaccinated that you know are eligible, making sure that when it's their turn, that people get vaccinated. That's going to be the step, especially with these variants that are popping up in South Africa, Brazil, some even here in California. You know, that certainly are working against the clock, and there's a lot of urgency in terms of, you know, how fast we can get people vaccinated. And so, you know, I encourage all of you, make sure you talk to your loved ones. People I know in the state, 65 and older are getting vaccinated. So make sure you talk to your grandparents, or, you know, grand aunts and uncles, and make sure, you know, assuage all those concerns that some people might have about the vaccine. And, you know, in the meantime, continue to stay safe, continue to avoid large gatherings. You know, I know this, we've been in this almost a year now. Hopefully we're getting close to real, you know, sustainable control of this pandemic. So just want to make sure that everyone is, you know, keeps doing their part. And like I said, is an advocate in terms of defeating this virus.
Thank you very much, Dr. Alexander. Dr. Leal?
Dr. Alexander gave a good advice for us all to focus on the people that are in the borderline, that they don't believe because they have information from both sides. So my advice to everybody else is that, okay, let's take this vaccine whenever they become available to us. And let's work on trying to convince these people that they are misinformed with all these negative ads and all these ideas that the vaccines are not good, that this is going to be good for everyone if a large part of the population gets vaccinated. And we don't want to talk about politics, but I would say to the politicians that, come on, guys, let's move on. Vaccines on the refrigerators do not do any good. They have to be on arms. And now we have been complaining that there was no vaccine, and then we didn't get much of vaccine. But here in the state of California, we have lots of people that want to get vaccinated, and we have lots of vaccines that have not been used yet. This is not acceptable in the same way that the lack of tests was not acceptable in the beginning. So let's keep up with the good work, and let's move on, and let's provide this vaccine for the larger part of the population, and for the students, that they keep informing the friends and members of the family and the others that may not believe in the vaccine, and to share the scientific side of it. Thank you very much for inviting me today.
Thank you, Dr. Leal. Ms. Hallock?
Yeah, I want to just thank you, everybody, for inviting me today. And I definitely am really fortunate, I would say, to be in the healthcare field. And I'm glad that I made the decision about 18 years ago to switch from economics into healthcare, because I think we're all in this together. And as much as I can do my part in it and spreading the word to people to please get vaccinated, I will do that. So thank you so much for having me today.
Thank you so much, Ms. Hallock. And finally, Dr. Haczku.
I also wanted to thank you for Alexander and everybody in the panels for very nice contributions. I just want to close with again 3 things. So get vaccinated as soon as you can. Keep on getting tested and wear masks. These are 3 basics, not so difficult to remember, but if we adhere to it, we can get rid of this nasty virus for good. And as Dr. Hallock said, we are in this together. So we need to work together to eliminate this threat, which is on all of us. It's almost like a global that like a sci-fi movie that we are living in, and we need to unite in order to get rid of this pandemic. So thank you
Right.
again.
Thank you. Well, thank you both. Thank you all four of you. We all thoroughly did enjoy the conversation, and I think I can speak on behalf of the students and say that we thoroughly learned a lot, and we appreciate all the time that you've given us. If students want to contact you all for whatever reason, is there some form of email or contact information you'd be able to put into the chat?
Yeah,
But
sure.
other than that, we appreciate your time. And thank you all so much for coming. Check us out for more events. And thank you to Dr. Haczku, Ms. Hallock, Dr. Leal, and Dr. Alexander once again.
You're welcome.
Thank you very much, Alex. Thank you for having me.
Thank you.